So here's something worth thinking about as you're lying there, settling in, letting the day fall away. Right now, in this exact moment, your brain is doing something extraordinary. It's changing, not in some vague metaphorical sense. I mean physically, structurally, chemically changing. The neurons in your head are adjusting their connections, strengthening some, weakening others, building tiny new bridges between cells that weren't linked an hour ago. And the wild part is, it's doing this because of everything that happened to you today. Every conversation you had, every problem you solved, every face you noticed in a crowd, every smell that drifted past you on the street, all of it left a mark. Microscopic, sure, but real. And by the time you fall asleep tonight, your brain will begin the quiet work of sorting through all of it, deciding what to keep, what to discard, and what to weave into the vast tapestry of everything you already know. That process, that constant, restless reshaping, is what we mean when we talk about learning. And honestly, it's one of the most fascinating things in all of science. Not because it's complicated, though it is, but because it's so deeply personal. Every single thing you know, every skill you have, every memory you carry, exists because your brain physically rewired itself to hold it. The way you recognize your mother's voice. The way your fingers know where the letters are on a keyboard without looking. The fact that you can understand these words right now, in real time, pulling meaning from a stream of sounds. All of that is the result of learning. And tonight, I want to walk you through how it actually works. Not the simplified version, the real thing. How your brain takes raw experience and turns it into knowledge, into skill, into you. But let's start at the beginning. And I mean the very beginning. Because people have been wondering about this for a very long time. If you'd asked an ancient Egyptian physician where thoughts came from, they probably would have pointed to the heart. That was the seat of intelligence as far as they were concerned. The brain? That spongy mass inside the skull? They didn't think much of it. When they mummified their dead, they actually scooped the brain out through the nose with a hook and threw it away. The heart was carefully preserved. The brain was garbage, which is kind of humbling when you think about it. The organ responsible for all of human civilization, and for thousands of years, we literally tossed it in the trash. The Greeks started to change that. Hippocrates, around 400 BC, was one of the first to argue that the brain was the organ of sensation and thought. He wrote that men ought to know that from the brain and from the brain only arise our pleasures, joys, laughter, and jests, as well as our sorrows, pains, griefs, and tears. That's a pretty bold claim for the time. Not everyone agreed. Aristotle, who came along a generation later, and who was wrong about a surprising number of things for someone so brilliant, went right back to the heart theory. He thought the brain was basically a cooling system for the blood, like a radiator, which, points aren't great for Aristotle on this one. But even the people who correctly identified the brain as the seat of the mind had no real idea how it worked, and you can't blame them. If you crack open a skull and look at a brain, there's nothing obvious happening. It doesn't move like a muscle. It doesn't pump like a heart. It just sits there, pinkish gray and wrinkled, looking for all the world like something inert. The idea that this quiet lump of tissue could be responsible for everything from composing symphonies to catching a ball, that took centuries to even begin to unravel. The first real breakthrough came from an unlikely place. In the late 1700s, an Italian physician named Luigi Galvani was dissecting frogs in his laboratory in Bologna. noticed something strange. When he touched a metal scalpel to the nerve of a dead frog's leg, the leg twitched, kicked, really, as if the frog were alive. Now, Galvani didn't fully understand what was happening, but he'd stumbled onto something enormous. He called it animal electricity, the idea that living tissue, and nerves in particular, operated through some kind of electrical force. He published his findings in 1791, and it caused a sensation. His nephew, Giovanni Aldini, took the idea even further, famously applying electrical current to the bodies of executed criminals in public demonstrations, making their jaws clench and their eyes open. It was macabre, it was theatrical, and it was, in a strange way, the birth of neuroscience. Because what Galvani had discovered, even if he didn't have the vocabulary for it yet, was that the brain communicates through electrical signals. That was the first clue. But knowing that the brain uses electricity doesn't tell you much about how it learns. For that, you need to understand the brain's basic architecture. And that part of the story belongs to a man named Santiago Ramon y Cajal. And honestly, his life is one of the great underdog stories in the history of science. Cajal was born in 1852 in a tiny village in Navarre in northern Spain. His father was a surgeon, practical and stern, and young Santiago was, by all accounts, a terrible student. Not because he wasn't smart, he was ferociously intelligent, but because he was restless, defiant, and obsessed with drawing. He wanted to be an artist. His father wanted him to be a doctor. They fought about it constantly. At one point, the young Cajal was actually thrown in jail for a minor act of rebellion. His father, trying to steer him toward medicine, apprenticed him to a barber and then a shoemaker, hoping manual labor would break his spirit. It didn't. Eventually, Cajal did study medicine, partly to please his father, partly because he realized that anatomy gave him something to draw. And that turned out to be the key to everything. Because Cajal wasn't just a good artist, he was a phenomenal one. He had an eye for detail that was almost superhuman. And when he encountered a new technology that was revolutionizing biology, a staining technique developed by an Italian scientist named Camillo Golgi, everything clicked. Here's what Golgi's stain did. The brain, under a normal microscope, is an almost impenetrable tangle. Billions of cells packed together so tightly that you can't make out individual structures. It's like trying to trace a single thread in a ball of yarn the size of a grapefruit. But Golgi's silver stain, for reasons that are still not entirely understood, only colors a tiny fraction of the cells, maybe 1 or 2%, and it colors them completely, from the cell body all the way out to the tips of their finest branches. Suddenly, individual brain cells popped out of the chaos like trees against a winter sky. Cajal took this technique and ran with it. He spent years hunched over his microscope, drawing what he saw with exquisite precision, and what he saw changed everything. He realized that the brain was not a continuous network, not a single connected web, the way Golgi himself believed. Instead, it was made up of individual, discrete cells, cells that came very close to each other but didn't actually touch. Each cell was its own unit, its own little processing station, sending signals to its neighbors across tiny gaps. Those cells we now call neurons, and those tiny gaps between them, those are synapses. And this is where the story of learning really begins. Think about what Cajal was proposing. The brain isn't a single mesh. It's a network of individual units, each one making specific connections to specific other units. And the pattern of those connections, which neurons talk to which other neurons, and how strongly, that pattern is what makes you, you. Your memories, your skills, your personality, your habits, all of it is encoded in the specific wiring diagram of your 100 billion neurons and their trillions of synapses. Kajal published his work throughout the 1890s and early 1900s, and in 1906 he shared the Nobel Prize in Physiology or Medicine with, of all people, Camillo Golgi, the very man whose staining technique he'd used, and who disagreed with him about what the stain revealed. Golgi still believed the brain was a continuous network. Kajal said it was made of discrete cells. They shared the Nobel, and reportedly, the ceremony was awkward. Golgi used his Nobel lecture to argue against Kajal's theory. Kajal used his to lay out the evidence for it. History sided with Kajal. But here's what Kajal also intuited, and this is remarkable. He suggested that learning might involve the strengthening of connections between neurons. That when you learn something, the synapses involved in that experience become more efficient, more robust. He didn't have the tools to prove it. He couldn't measure synaptic strength or watch connections change in real time. But he looked at those beautiful branching neurons under his microscope, and he thought, the secret must be in the connections, not in the cells themselves, but in how they talk to each other. He was right, and it would take another 50 years for someone to formalize that intuition into a real theory. That someone was a Canadian psychologist named Donald Hebb, and in 1949, he published a book called The Organization of Behavior that quietly changed the course of neuroscience. Hebb wasn't a bench scientist peering through microscopes. He was a theorist, a thinker, someone who looked at the available evidence and tried to build a coherent framework for how the brain could possibly learn. And his central idea was beautifully simple. He proposed that when two neurons fire at the same time, repeatedly, the connection between them gets stronger. The synapse linking them becomes more efficient, so that in the future, when one fires, the other is more likely to fire too. The shorthand version, which neuroscience students learn on day one, is neurons that fire together, wire together. Now, that's a catchy phrase, and it captures the essence, but let's slow down and think about what it actually means. Imagine you're a baby, and you see your mother's face. At the same moment, you hear her voice. The neurons in your visual cortex that respond to her face are firing. The neurons in your auditory cortex that respond to her voice are firing. And because they're firing at the same time, over and over, day after day, the connections between those two groups of neurons get stronger. Eventually, just hearing her voice activates a faint echo of her face in your mind's eye. Just seeing her face activates the memory of how she sounds. The two have become linked. That's an association. That's a memory being born. Hebb's rule is elegant because it doesn't require a teacher. It doesn't require someone to come in and manually wire the brain. It's automatic. It emerges from the statistics of experience. Whatever happens together in the world gets linked together in the brain. The brain is, in a very real sense, a machine for detecting correlations in the environment and encoding them into its own structure. But Hebb was working in 1949. He had no way to directly test his theory at the level of individual synapses. He couldn't watch two neurons strengthen their connection in real time. He could only reason about it. And so for decades, Hebb's rule remained an elegant hypothesis, widely believed, deeply influential, but unproven. The proof would come from an unexpected creature, in an unexpected laboratory, through an experiment that almost didn't work. And the two scientists who pulled it off would spend years refining their technique before they finally caught the brain in the act of learning. Their names were Tim Bliss and Terje Lomo, and the creature was a rabbit. Now, to understand what they did, you need to picture where they were working. It was the early 1970s, and they were in Per Anderson's laboratory in Oslo, Norway. Anderson was one of the leading neurophysiologists of his day, and his lab had become a magnet for young scientists interested in the hippocampus, that seahorse-shaped structure tucked deep inside the temporal lobe that we now know is absolutely critical for forming new memories. At the time, people were beginning to suspect the hippocampus was important, but the full picture was still emerging. What made it attractive to experimentalists was its anatomy. The hippocampus has a beautifully organized, layered structure. The neurons are arranged in neat, well-defined pathways, almost like lanes on a highway. You can stimulate one group of neurons and record from another group downstream, and you know exactly which pathway you're activating. It's unusually clean for brain tissue. And that mattered enormously, because what Bliss and Lomo wanted to do required precision. Here's what they were after. They wanted to test Hebb's idea directly. If neurons that fire together really do wire together, then it should be possible to artificially make a group of neurons fire intensely and then check whether the synaptic connections downstream got stronger as a result. Simple in concept, brutally difficult in practice. Terje Lomo was a Norwegian PhD student, quiet and meticulous, and he had actually stumbled onto something strange during his earlier experiments. He'd been stimulating a pathway in the hippocampus of anesthetized rabbits, specifically a bundle of fibers called the perforant path, which sends signals into a region called the dentate gyrus. He'd deliver a brief burst of high-frequency electrical pulses, maybe 100 pulses in 1 second, and then go back to his normal, gentle test pulses to see how the downstream neurons responded. And what he noticed was that after that intense burst, the response got bigger. The downstream neurons were reacting more strongly to the same gentle stimulus than they had before. It was as if the synapses had been turned up, like someone had nudged the volume knob. And here's the thing that really caught his attention. It lasted. It didn't fade after a few seconds or a few minutes. The enhancement persisted for hours. In some cases, as long as he kept recording, the effect was still there. Lomo mentioned this to Bliss, a British postdoctoral researcher who had come to Oslo specifically to learn hippocampal electrophysiology. Bliss immediately recognized the significance. If this was real, if you could genuinely induce a long-lasting strengthening of synaptic connections just by making neurons fire together vigorously, then you were looking at a physical mechanism for memory. You were watching Hebb's rule play out in real tissue. But they had to be careful. The scientific community would be skeptical. There were 100 ways an artifact could creep into an experiment like this: electrode drift, changes in the rabbit's brain temperature, fluctuations in anesthesia depth. They spent years refining the technique, controlling for every variable they could think of. They repeated the experiment over and over. And finally, in 1973, they published their landmark paper in the Journal of Physiology. They called the phenomenon long-term potentiation, LTP for short. And that paper changed everything. Let's talk about what LTP actually is, because it's one of those ideas that sounds technical, but is really quite intuitive once you unpack it. Potentiation just means strengthening. Long-term means it lasts. So long-term potentiation is a long-lasting strengthening of the connection between two neurons after they've been activated together intensely. That's it. That's the core idea. When a synapse undergoes LTP, the sending neuron doesn't have to shout any louder. the receiving neuron just becomes more sensitive to the signal. It's like the difference between whispering to someone across a noisy room and whispering to someone who's leaning in close with their ear turned toward you. The message is the same, but the reception is dramatically better. Now, you might be thinking, okay, but how? What actually changes at the synapse to make this happen? And this is where it gets really beautiful, because the molecular machinery behind LTP is one of the most elegant systems in all of biology. Let me walk you through it, slowly, because it's worth understanding. At a typical excitatory synapse in the hippocampus, the sending neuron releases a chemical messenger called glutamate. Glutamate floats across that tiny synaptic gap. Remember Cajal's gap, the one he saw with his stained slides, and lands on receptors embedded in the membrane of the receiving neuron. Now, there are several types of glutamate receptors, but two of them are the main characters in this story. The first is called the AMPA receptor. Think of AMPA receptors as the workhorses of synaptic transmission. When glutamate hits an AMPA receptor, it opens a channel that lets positively charged ions flow into the receiving neuron, nudging it a tiny bit closer to firing. This is normal, everyday synaptic communication. It happens billions of times a second across your brain right now, even as you're lying there listening to this. The second type is the NMDA receptor, and this one is special. The NMDA receptor is what scientists sometimes call a coincidence detector, and that phrase should remind you of something. Hebb's rule, right? Neurons that fire together wire together. The NMDA receptor is the molecular embodiment of that rule, and here's why. Under normal conditions, the NMDA receptor is blocked. Even when glutamate binds to it, the channel doesn't open, because there's a magnesium ion physically sitting in the channel pore like a cork in a bottle. The only way to pop that cork out is if the receiving neuron is already partially depolarized, meaning it's already somewhat excited, already receiving a lot of input. So the NMDA receptor only opens when two things happen at the same time. Glutamate arrives from the sending neuron, and the receiving neuron is already active. It requires coincidence. It requires both sides of the synapse to be engaged simultaneously. Think about what that means. The NMDA receptor is essentially asking a question. Is this synapse part of a meaningful pattern? Is the signal arriving here correlated with activity that's already happening in this neuron? If the answer is no, if the sending neuron fires, but the receiving neuron is quiet, the magnesium plug stays in place and nothing special happens. But if the answer is yes, if both neurons are active at the same time, just like Hebb predicted, then the magnesium pops out, the NMDA channel opens, and calcium ions come flooding in. And calcium, in this context, is the trigger for everything that follows. Calcium is one of the most important signaling molecules in all of cell biology. When calcium levels rise inside the receiving neuron, it sets off a cascade of biochemical events. Enzymes are activated, proteins are modified, and one of the most important immediate effects is that more AMPA receptors get trafficked to the surface of the synapse. Remember, AMPA receptors are the workhorses. They're the ones that carry the everyday signal. So by inserting more of them into the membrane, the synapse becomes more sensitive to future signals. The next time the sending neuron releases glutamate, there are more AMPA receptors waiting to catch it. The synapse has been strengthened. It has been potentiated. And if the calcium signal is strong enough and sustained enough, it can trigger changes in gene expression back in the nucleus of the cell, leading to the construction of entirely new proteins that physically remodel the synapse, making it larger, adding new connection points, even growing new synaptic structures. This is the transition from short-term to long-term memory at the molecular level. Early LTP, lasting minutes to hours, depends on modifying existing proteins. Late LTP, lasting hours to days to potentially a lifetime, requires new protein synthesis. It requires the neuron to actually build new cellular machinery. And this is where you can start to appreciate something profound. When you learn something, really learn it, not just hold it in mind for a few seconds, but genuinely encode it into long-term memory. Your brain is physically constructing new biological structures. Proteins are being synthesized. Synapses are being remodeled. The shape of your neural connections is literally, physically different after you've learned something than it was before. Your brain tonight is not the same brain it was this morning. Every conversation you had, every new face you noticed, every problem you worked through, each one left a physical trace in the architecture of your synapses. That's not a metaphor. That's molecular biology. Now, Bliss and Lomo's 1973 paper didn't explain all of this molecular detail. That understanding would take decades of additional work by hundreds of laboratories around the world. The role of the NMDA receptor was worked out in the 1980s, largely through the efforts of Graham Collingridge in the UK, who showed that blocking NMDA receptors prevented the induction of LTP without affecting normal synaptic transmission. That was a crucial experiment because it separated the mechanism for learning from the mechanism for ordinary signaling. The brain uses one system for routine communication and a different, gated system for deciding when to strengthen a connection. It's like having a regular phone line for everyday calls and a special secure line that only activates when both parties pick up at the same time. The work on calcium's role, on the enzyme cascades, on the trafficking of AMPA receptors, all of that came from dozens of research groups through the 80s and 90s. Eric Kandel, whom we'll come back to later, was doing parallel work in a very different animal that would beautifully complement the LTP story. Richard Morris in Edinburgh developed the water maze, a deceptively simple behavioral test where rats have to learn the location of a hidden platform in a pool of opaque water. Morris showed that blocking NMDA receptors in the hippocampus didn't just prevent LTP in a dish. It prevented rats from forming spatial memories in real life. The animals could swim fine. They were motivated. They weren't impaired in any obvious way, but they couldn't learn where the platform was. That was a powerful link between the molecular mechanism and actual behavior. It suggested that LTP wasn't just a laboratory curiosity. It was the real thing, the actual process the brain uses to encode new information. But here's something important to keep in mind. The brain doesn't just need to strengthen connections. If all synapses could do was get stronger and stronger, eventually everything would be maxed out. The system would saturate. You'd lose the ability to distinguish between important signals and noise. It would be like trying to highlight the key passages in a book by highlighting every single line. The highlighting becomes meaningless. So the brain needs a counterbalancing process, a way to weaken connections that are no longer relevant. And sure enough, a few years after LTP was established, researchers discovered its mirror image, long-term depression, or LTD. And no, it has nothing to do with the mood disorder. It's a technical term meaning a long-lasting decrease in synaptic strength. LTD tends to occur when the timing between neurons is slightly off, when the correlation is weak, or the firing is out of sync. Low-frequency stimulation, slow and desultory, rather than the intense bursts that trigger LTP, tends to weaken synapses rather than strengthen them. And this pairing, LTP and LTD, strengthening and weakening, signal and sculpting, gives the brain an extraordinary flexibility. It's not just adding connections, it's constantly adjusting them, turning some up and others down, refining the pattern of which neurons are linked to which. Think of it less like writing in ink and more like working in clay. You're always shaping, always molding, pressing in here and smoothing out there. The sculpture is never quite finished. This brings us to something that puzzled researchers for a long time, something that seems almost paradoxical when you first hear it. Because the hippocampus, the structure where Bliss and Lomo discovered LTP, the structure that's so essential for forming new memories, it turns out the hippocampus doesn't actually store most of your long-term memories permanently. It's more like a temporary workspace, a staging area. Memories form there, but over time, they migrate. They get transferred to other parts of the brain, particularly the neocortex, that vast wrinkled outer layer that makes up most of what you see when you look at a brain. This process is called memory consolidation, and one of the most fascinating things about it is when it happens, because a huge amount of consolidation occurs while you're sleeping. Right now, in fact, if you're starting to drift off, your brain may already be beginning that extraordinary process. So let's talk about what's actually happening in there when the lights go out. For a long time, sleep was considered essentially downtime. The brain was resting, recuperating, maybe doing some basic housekeeping. And from the outside, that's what it looks like. You're lying still. Your muscles are relaxed. Your eyes are closed. But when researchers started recording brain activity during sleep, starting seriously in the 1950s with Nathaniel Kleitman and his graduate student Eugene Aserinsky at the University of Chicago, they discovered something that genuinely surprised them. The sleeping brain isn't quiet at all. In some phases of sleep, it's almost as electrically active as when you're wide awake. Aserinsky noticed that sleeping subjects would periodically move their eyes rapidly beneath their closed lids, rapid eye movement, REM, and when he woke people during these phases, they almost always reported vivid dreams. But what's really interesting for our purposes isn't the dreaming itself. It's what's happening at the level of neurons and synapses during the different stages of sleep, because it turns out the brain is doing something remarkably purposeful. Here's the picture that's emerged over the last few decades, and it's genuinely beautiful in its logic. During the day, as you go about your life, learning someone's name, navigating a new route, figuring out how to use a new app, absorbing the emotional texture of a conversation, the hippocampus is rapidly encoding these experiences. It's creating these initial memory traces using the LTP mechanisms we talked about, strengthening certain synaptic connections, forming temporary patterns. But the hippocampus has limited capacity. It's a fast learner but a small notebook. If everything stayed there permanently, you'd eventually run out of space, or more accurately, new memories would start interfering with old ones. The hippocampus needs to be cleared out, refreshed, made ready for the next day's experiences. So what happens during sleep, particularly during the deep slow wave stages that dominate the first half of your night, is a kind of replay. And I mean that almost literally. Researchers have recorded from individual neurons in rats. This work was pioneered by Matthew Wilson and Bruce McNaughton at the University of Arizona in the mid-1990s, and what they found was extraordinary. They'd have rats run through a maze during the day, and they'd record the firing patterns of place cells in the hippocampus. Place cells are neurons that fire when the animal is in a specific location, and they were discovered by John O'Keefe in the 1970s, which eventually won him a Nobel Prize in 2014. So each location in the maze has its own little constellation of cells that light up. And when Wilson and McNaughton recorded from those same cells while the rats slept afterward, the cells replayed the same sequences, the same patterns in the same order, as if the rat were running the maze again in its sleep. But here's what's really clever. The replay happened faster, compressed in time, like someone fast-forwarding through a recording. And during this replay, something critical is happening. The hippocampus is essentially teaching the neocortex. It's replaying the memory trace over and over, and each time it does, it's strengthening the corresponding connections in the cortical networks. Slowly, gradually, the neocortex builds its own version of the memory, one that's more stable, more integrated with everything else you already know. Think of the hippocampus as a journalist who witnessed the event firsthand and is now dictating the story to an archivist who's going to file it permanently. The journalist tells the story multiple times, each time helping the archivist get the details right, make the right cross-references, file it in the right place. And eventually, the archivist has a solid enough copy that the journalist's notes can be recycled. This is why sleep deprivation is so devastating to learning. It's not just that you're tired and can't pay attention, although that's certainly part of it. It's that you're literally cutting short the consolidation process. The hippocampus doesn't get to finish its replay. The neocortex doesn't get to build those stable long-term traces. The memories are left fragile, half-formed, much more likely to fade. There's a study from 2000 by Robert Stickgold and colleagues at Harvard that demonstrated this beautifully. They trained subjects on a visual discrimination task, basically learning to spot subtle patterns, and tested them later. Subjects who slept normally after training showed significant improvement when retested. Subjects who were sleep-deprived the first night after training, even if they got plenty of recovery sleep afterward, showed no improvement. The window for consolidation had closed. That first night of sleep after learning something new is disproportionately important. Now, I should mention that this two-stage model of memory, fast hippocampal learning followed by slow cortical consolidation, was actually proposed theoretically before a lot of the experimental evidence came in. David Marr, the brilliant British neuroscientist, who we should really talk about more, sketched out something like this idea back in 1971. Marr died tragically young, at just 35, from leukemia in 1980. But in his short career, he made contributions to understanding the hippocampus, the cerebellum, and visual processing that were decades ahead of their time. His idea was that the hippocampus performed a kind of simple, fast initial storage, and that memories were later reorganized and transferred to the neocortex during offline periods. He didn't have the experimental tools to prove it, but the basic architecture he proposed has held up remarkably well. And then there's the work that tied all of this together at the molecular level, which brings us back to someone I mentioned earlier, Eric Kandel. If Cajal is the father of modern neuroscience and Hebb is the prophet of synaptic learning, then Kandel is the person who proved the prophecy true, down to the level of individual molecules. And he did it in one of the most unlikely model organisms you could imagine. Kandel is a Viennese-born American neuroscientist who fled Austria as a child in 1939, just after Kristallnacht. His family was Jewish, and the experience of that upheaval, of watching a cultured society transform into something monstrous left him with a deep, lifelong fascination with memory. How do people remember? How do experiences change us? He started in psychoanalysis actually, trained as a psychiatrist, was deeply interested in Freud, but gradually became convinced that to really understand memory, you had to understand it at the level of cells and molecules. You had to get your hands on the actual biological machinery. And here's where the choice of organism matters so much. The human brain has roughly 86 billion neurons. A rat's brain has a few hundred million. Even a fruit fly has about 100,000. Kandel wanted something simpler, much simpler. He chose Aplysia californica, the California sea slug. A marine snail, basically. Its entire nervous system contains about 20,000 neurons, and some of those neurons are enormous, visible to the naked eye, big enough to impale with an electrode without a microscope. You could identify individual cells, return to the same cell day after day, and watch what happened to it as the animal learned. Now, you might reasonably wonder what a sea slug could possibly teach us about human learning. And that skepticism was widespread when Kandel started this work in the 1960s. Plenty of serious neuroscientists thought he was wasting his time, but Kandel had an insight that turned out to be profoundly correct. The basic molecular mechanisms of learning are conserved across evolution. The same fundamental processes that strengthen or weaken synapses in a sea slug also operate in your brain. Evolution doesn't reinvent the wheel. Once it finds a molecular trick that works for adjusting synaptic strength, it keeps using it, elaborating on it, building more complex systems on top of it, but the foundation stays remarkably similar. What Kandel studied in Aplysia was a simple defensive reflex. The sea slug has a siphon, a little fleshy tube it uses for breathing, and if you touch the siphon gently, the animal withdraws its gill. A protective reflex, like pulling your hand back from something hot. And this reflex can be modified by experience in ways that look a lot like basic forms of learning. If you touch the siphon repeatedly and nothing bad happens, the animal gradually stops responding. The gill withdrawal gets weaker and weaker. This is habituation, the simplest form of learning, the kind that lets you tune out the ticking clock or the hum of your refrigerator. At the synaptic level, Kandel showed that habituation involves a decrease in the amount of neurotransmitter released at the synapse between the sensory neuron and the motor neuron. The connection gets quieter. Fewer vesicles of neurotransmitter fuse with the membrane each time the signal comes through. But if you pair the gentle touch with a sharp shock to the tail, something aversive, something that matters, the reflex gets dramatically stronger. The animal becomes sensitized. Now even a light touch produces a vigorous gill withdrawal. And at the molecular level, Kandel and his colleagues traced out the entire cascade. The shock activates modulatory interneurons that release serotonin onto the sensory neurons' terminals. The serotonin triggers a second messenger cascade involving cyclic AMP and protein kinase A, which ultimately increases neurotransmitter release. The synapse gets louder. More glutamate pours out with each signal. Here's the thing that really matters, the part that connects to everything we've been talking about. Kandel found that short-term sensitization, lasting minutes to hours, involves only these chemical modifications. The existing proteins get tweaked. The existing machinery gets turned up. But long-term sensitization, lasting days or even weeks, requires something more. It requires new protein synthesis. It requires gene expression. The protein kinase A actually travels into the nucleus of the cell, activates a transcription factor called CREB, and CREB switches on genes that produce new proteins. These proteins travel back to the synapse and physically remodel it. New synaptic connections actually grow. The neuron sprouts new terminals. Think about what that means. When you learn something and remember it for the long term, genes in your neurons are being activated. New proteins are being built. The physical structure of your brain changes. Learning isn't just a pattern of electrical activity, it's a construction project. And Kandel showed this first in a sea slug with a clarity and molecular precision that was breathtaking. He won the Nobel Prize in 2000 for this work, and it's one of those cases where the prize was, if anything, overdue. And what's remarkable is how well his findings mapped onto what other researchers were discovering about LTP in mammalian brains. Remember the distinction between early LTP and late LTP? Early LTP involves modifying existing proteins and trafficking more AMPA receptors to the synapse. That's like Kandel's short-term sensitization. Late LTP requires new protein synthesis, new gene expression, structural remodeling. That's like his long-term sensitization. The parallels are striking, and they're not coincidental. They reflect a deep evolutionary conservation of the molecular logic of learning. So now we have a picture that's getting richer and more detailed. We know that learning involves changes in synaptic strength. We know the molecular machinery. NMDA receptors as coincidence detectors, calcium cascades, CREB, and gene expression, structural remodeling. We know that the hippocampus is a fast initial learner and that memories consolidate into the neocortex during sleep. We know that these mechanisms are ancient, shared across species from sea slugs to humans. But there's a whole dimension of learning we haven't touched yet, one that takes us beyond individual synapses and into the question of how the brain organizes itself on a larger scale. Because your brain doesn't just strengthen random connections, it builds maps. It creates organized representations of the world, maps of space, maps of sound frequencies, maps of your own body. And the way those maps form, especially early in life, involves a process that is both astonishing and, when it goes wrong, heartbreaking. It's the story of critical periods, and it starts with a pair of researchers who spent years doing something that sounds almost absurdly simple. They sewed shut the eyes of kittens. David Hubel and Torsten Wiesel, working at Johns Hopkins and later Harvard, starting in the late 1950s and continuing through the 1960s and 70s. Their experiments were deceptively straightforward. They wanted to understand how the visual cortex develops, how the brain learns to see. And the way they approached it changed everything we know about how experience sculpts the developing brain. Here's what they did. They took young kittens, just a few days or weeks old, and surgically sutured one eyelid shut. Not both eyes, just one. The kitten could still see perfectly well out of the other eye. They left the sutures in place for a specific period of time, sometimes weeks, sometimes months. Then they opened the eye back up and tested what had happened to the visual cortex. And what they found was devastating in its clarity. The eye itself was fine. The retina was healthy. The optic nerve was intact. Light could enter and signals could travel. But the kitten was functionally blind in that eye. Not because anything was wrong with the eye, but because the brain had never learned to use it. The neurons in the visual cortex that should have responded to input from that eye had been taken over, colonized, essentially, by the open eye. The brain had reorganized itself based on experience, and the closed eye had lost its territory permanently. Now, here's where it gets really interesting. If they did the exact same thing to an adult cat, sewed one eye shut for the same amount of time, nothing happened. They'd open the eye, and the cat could see just fine. The visual cortex hadn't reorganized at all, which meant there was something special about that early period of life. There was a window of time during which experience didn't just influence the visual cortex, it was required. Without the right input during that window, the wiring never formed properly, and once the window closed, it was too late. Hubel and Wiesel called this a critical period, and the concept turned out to be one of the most important ideas in all of neuroscience. They mapped it carefully. In cats, the critical period for visual cortex development peaks at around 4 to 6 weeks of age and gradually closes over the next few months. During that window, the brain is astonishingly plastic. It's being shaped by whatever input it receives. After the window closes, the basic architecture is largely locked in. Think about what that means. It means the brain isn't just passively recording experience. During critical periods, experience is literally building the hardware. It's like the difference between writing software for a computer and actually wiring the circuits of the computer itself. During a critical period, the input is determining the physical structure of the neural networks. That's a fundamentally different kind of learning than what we talked about earlier with LTP and synaptic strengthening. This is large-scale architectural construction, and the mechanism behind it is beautiful. In the visual cortex, neurons are organized into what Hubel and Wiesel called ocular dominance columns, alternating stripes of neurons, some primarily driven by the left eye, some by the right. In a normal brain, these columns are roughly equal in width. Each eye gets its fair share of cortical real estate. But when one eye is deprived during the critical period, the columns for the open eye expand dramatically, and the columns for the closed eye shrink to almost nothing. The neurons are competing for territory, and the eye that's providing input wins. This is competitive plasticity, and it's driven by the same Hebbian principles we talked about earlier. Neurons that fire together wire together. The open eye is sending correlated, meaningful signals to the cortex. Those signals drive postsynaptic neurons to fire, the synapses get strengthened, and those connections stabilize and expand. The closed eye is sending nothing, or at best random noise. Its synapses weaken and get pruned away, its Heb's rule playing out on a massive scale, shaping the entire organization of a brain region. Hubel and Wiesel won the Nobel Prize in 1981 for this work, sharing it with Roger Sperry, and it had immediate and profound clinical implications. Pediatric ophthalmologists had long known about a condition called amblyopia, what people casually call a lazy eye. A child might have one eye that's slightly misaligned, or has a cataract, or is just weaker than the other. If you catch it early enough and treat it, by patching the strong eye, forcing the brain to use the weak one, you can often restore normal vision. But if you wait too long, the treatment doesn't work. And now, thanks to Hubel and Wiesel, doctors understood why. The critical period had closed. The cortical territory had been permanently reassigned. This is one of those places where basic science research on cats directly saved children's vision. The urgency of early screening for childhood eye problems, the practice of patching, the understanding that you have a limited window, all of that flows from those experiments in Hubel and Wiesel's lab. But critical periods aren't just about vision. They exist throughout the brain for all sorts of functions, and they open and close on different schedules. Language is probably the most famous example. You already know this intuitively. Children learn languages effortlessly. They absorb grammar, pronunciation, vocabulary. They just soak it up like sponges. But try learning a new language as an adult, and it's brutal. You can do it, sure, But it's slow, it's effortful, and you'll almost certainly never sound like a native speaker. The linguist Eric Lenneberg proposed in 1967 that there's a critical period for language acquisition, and decades of research have largely confirmed this. The window seems to be open widest in the first few years of life and gradually narrows through puberty. The evidence comes from many sources. Studies of deaf children who received cochlear implants at different ages, studies of feral children tragically deprived of language exposure, studies of immigrant families where the children become fluent, but the parents always retain an accent. The pattern is remarkably consistent. Earlier exposure leads to better outcomes, and there seems to be a point after which native-level acquisition becomes nearly impossible. Now, here's something that might be keeping you wondering. If critical periods are so important, and if they close, does that mean the adult brain can't really learn? Are we stuck with whatever wiring we ended up with by age 12 or 15? And the answer is no, not at all. But the kind of learning that happens in adulthood is genuinely different from what happens during critical periods. During a critical period, the brain is doing large-scale construction. It's deciding which neural circuits to build and which to prune away. It's establishing the basic architecture. After the critical period closes, the brain can still learn. It can still strengthen and weaken synapses, still form new memories, still acquire new skills. But it's working within the framework that was established during those early years. It's like the difference between building a house and redecorating it. You can change the furniture, repaint the walls, even knock out a non-load-bearing wall here and there. But the foundation, the framing, the basic floor plan, that was set during construction. And there's a molecular reason for this. During critical periods, the balance between excitatory and inhibitory neurotransmission is tilted in a way that promotes plasticity. As the critical period closes, inhibitory circuits, particularly those involving a neurotransmitter called GABA, mature and strengthen, essentially putting the brakes on large-scale reorganization. There are also physical structures that form around certain neurons, called paraneuronal nets, which are like a kind of molecular scaffolding that stabilizes the existing connections and makes them resistant to change. It's as if the brain deliberately locks down its own wiring once the critical period is over, protecting what it's built from being disrupted by new experience. This has led to one of the most exciting frontiers in modern neuroscience: the question of whether you can reopen critical periods. If you could dissolve those paraneuronal nets, or shift the excitatory-inhibitory balance back toward its juvenile state, could you restore the brain's youthful plasticity? There have been tantalizing results in animal studies. Researchers have used enzymes to dissolve paraneuronal nets in adult rats and found that it does restore some degree of critical period-like plasticity. Others have found that certain drugs, like valproic acid, seem to partially reopen the critical period for things like absolute pitch in adults. The work is preliminary, and we're a long way from clinical applications, but the idea that the adult brain's limitations might not be permanent, that's genuinely thrilling. But let's pull back from the molecular level for a moment, because there's a different kind of learning story happening in the brain that operates on a completely different time scale, and it's one you experience every single day without thinking about it. It's the story of how you learn from reward and punishment, how you figure out which actions lead to good outcomes and which ones lead to bad ones, how you learn to make decisions. And this story centers on one of the most important molecules in your brain: dopamine. You've probably heard of dopamine. It gets called the pleasure chemical in pop science articles, and that's not exactly wrong, but it's so incomplete that it's almost misleading. Dopamine is really about something more subtle and more powerful than pleasure. It's about prediction. In the early 1990s, a neuroscientist named Wolfram Schultz was recording from dopamine neurons in the brains of monkeys. These neurons sit deep in the midbrain, in a region called the ventral tegmental area, and they project widely throughout the brain, sending dopamine to the prefrontal cortex, the striatum, and other regions involved in decision-making and motivation. Schultz was watching what happened to these neurons as monkeys learned simple tasks. Press a lever, get a squirt of juice. At first, when the monkey received the juice unexpectedly, the dopamine neurons fired a burst of activity. That makes sense. It's a reward, and the brain is registering it. But here's what happened as the monkey learned. Schultz trained the monkey to associate a light or a tone with the upcoming juice reward, and as the monkey learned the association, something remarkable shifted. The dopamine neurons stopped firing when the juice arrived. Instead, they fired when the predictive cue appeared. The light. The tone. The signal had moved backward in time, from the reward itself to the predictor of the reward. And here's the kicker. If the cue appeared, but the juice didn't come, if the expected reward was omitted, the dopamine neurons didn't just stay quiet, they actively decreased their firing rate below baseline at the exact moment the juice should have arrived. A dip. A negative signal. The brain wasn't just tracking rewards. It was tracking the difference between expected rewards and actual rewards. It was computing what neuroscientists call a reward prediction error. This was a revelation, and it connected beautifully to a concept from computer science and artificial intelligence called temporal difference learning, which had been developed by Richard Sutton and Andrew Bardo in the 1980s. The basic idea is that you don't need to wait until the end of an experience to learn from it. You can learn at every step by comparing what you expected to happen with what actually happened. If things turn out better than expected, you strengthen whatever behavior led to that outcome. If things turn out worse than expected, you weaken it. And Schultz's dopamine neurons were doing exactly this. They were broadcasting a prediction error signal to the rest of the brain, a signal that could be used to update behavior in real time. This is how you learn from experience in the most practical, everyday sense. It's why the first bite of an amazing meal is the most exciting. Your brain didn't predict it would be that good, so there's a big positive prediction error, a surge of dopamine. By the 10th bite, your brain has updated its predictions, and the dopamine response is smaller. It's why unexpected gifts feel so much better than expected ones. It's why the anticipation of something good can feel almost as rewarding as the thing itself, because the dopamine has shifted to the anticipatory cue. And it's also why addiction is so devastating, because drugs of abuse hijack this system. They flood the brain with dopamine in ways that dwarf any natural reward, creating prediction errors so large that the learning system goes haywire. The brain learns, with tremendous force, that whatever preceded the drug is the most important predictor in the world, and that learning is encoded in the same synaptic machinery we've been talking about, LTP, in the dopamine circuits of the striatum, NMDA receptors, calcium cascades, structural remodeling. The same mechanisms that let you learn to ride a bicycle or remember your first kiss are the ones that make addiction so stubbornly, heartbreakingly hard to unlearn. The synapses have been rewritten, the circuits have been physically reshaped, and every cue associated with the drug, a particular street corner, a certain time of day, even a mood, has been stamped into the brain's prediction machinery with a force that no natural experience can match. That's not a moral failing. That's neuroscience. That's the learning system doing exactly what it evolved to do, just pointed at something that destroys the person it's trying to help. And here's what makes it even more insidious. Remember how Schultz showed that dopamine shifts from the reward itself to the cue that predicts the reward? In addiction, that shift becomes extreme. The sight of paraphernalia, the smell of a particular environment, even a subtle emotional state. These cues start triggering massive dopamine responses, creating intense craving before the drug is anywhere in sight. Meanwhile, the drug itself often stops producing much pleasure at all, because the brain has adjusted its baseline. Tolerance builds. The person needs more of the substance just to feel normal. But the cues, the predictors, those keep screaming. The wanting becomes completely divorced from the liking. You can desperately want something you don't even enjoy anymore. That distinction between wanting and liking was beautifully teased apart by a neuroscientist named Kent Berridge at the University of Michigan, starting in the late 1980s and continuing through the 1990s and 2000s. Berridge showed that dopamine is really about wanting, about incentive salience, he called it, not about pleasure itself. Pleasure, the actual hedonic experience, seems to depend on a different and much smaller set of brain circuits involving opioid and endocannabinoid signaling in tiny hotspots within structures like the nucleus accumbens and the ventral pallidum. You can knock out dopamine in a rat, and it will still show pleasure reactions when you put sugar on its tongue. It'll still lick its lips, still show the facial expressions of enjoyment. But it won't work to get the sugar. It won't cross a room. It won't press a lever. The wanting is gone, even though the liking remains. And in addiction, you get the opposite dissociation: enormous wanting, diminished liking. The learning system has been captured. So let's sit with that for a moment, because it tells us something profound about what learning really is at the neural level. Learning isn't just the passive recording of experience. It's an active process of prediction and correction, driven by specific chemical signals, encoded in specific synaptic changes, and shaped by the value the brain assigns to outcomes. Dopamine doesn't just say, "That was good." It says, "That was better or worse than expected. And here's how much you should update your model of the world." And that updating happens through the same molecular machinery: the NMDA receptors, the calcium influx, the CREB activation, the protein synthesis, the growth of new dendritic spines, that we traced all the way back to Kendall's sea slugs and Bliss and Lomo's rabbit hippocampi. It's one system. Different brain regions, different circuits, different behavioral purposes, but the fundamental logic of synaptic plasticity runs through all of it like a common language. Now, I want to take you somewhere slightly different, because there's a whole dimension of learning we haven't really explored yet, and it's one that might be the most relevant to your everyday life. We've talked about how individual synapses get stronger or weaker. We've talked about how sleep consolidates those changes. We've talked about critical periods when the brain is especially plastic, and about how dopamine steers learning toward valuable outcomes. But what about learning a skill? Not memorizing a fact, not forming an emotional association, but actually getting better at something. Playing piano, throwing a ball, typing on a keyboard, speaking a second language fluently. Motor learning, procedural learning, skill acquisition. This is the kind of learning where you practice something over and over, and you're terrible at first, and then gradually, almost imperceptibly, you get better. And the neural story here is fascinating because it involves a different set of brain structures working together in a way that shifts over time. The key player in early skill learning is once again the prefrontal cortex, that same region we've touched on as the seat of working memory and executive control. When you first try to learn a new skill, your prefrontal cortex is working overtime. Think about the first time you tried to drive a car with a manual transmission. You had to consciously think about every single step. Press the clutch, shift the gear, ease off the clutch while pressing the gas, check the mirror, steer. And all of it felt impossibly demanding, like juggling while reciting poetry. That's your prefrontal cortex and associated areas trying to coordinate a sequence of actions that haven't been automated yet. Every movement requires deliberate attention. Your brain is essentially problem-solving its way through each moment. But here's what's really interesting. As you practice, as you repeat the sequence over and over, control of that behavior gradually transfers from the prefrontal cortex and associated cortical areas to the basal ganglia, particularly the striatum, the same structure we were just talking about in the context of reward learning and addiction. The striatum turns out to be critical for chunking sequences of actions into smooth, automatic routines. Anne Graybiel at MIT has done some of the most beautiful work on this, starting in the 1990s and continuing for decades. She recorded from neurons in the striatum of rats as they learned to navigate mazes for a chocolate reward. And what she found was remarkable. Early in learning, striatal neurons fired throughout the entire maze run. They were active at every turn, every decision point, tracking the whole sequence moment by moment. But as the rats became expert, as the behavior became habitual, the firing pattern changed dramatically. The neurons started firing mainly at the beginning and end of the sequence, at the start of the maze run, and at the moment of reward. The middle of the sequence, all those individual turns and decisions, became chunked into a single automated unit. The chi striatum had essentially packaged the whole behavior into one routine that could be triggered by a start cue and run to completion without conscious oversight. Think about what that means for your own life. When you first learned to type, you hunted for each key individually. Now your fingers fly across the keyboard and you're not thinking about the location of any letter. You're thinking about the words, the ideas. The individual keystrokes have been chunked into automatic motor programs stored in your basal ganglia. When you drive a familiar route to work, you sometimes arrive and realize you don't remember the drive at all. That's because the whole sequence has been handed off from conscious cortical control to automatic striatal control. Your prefrontal cortex was free to think about other things, your grocery list, a conversation from yesterday, because the driving had become a habit. And this transfer from cortical to subcortical control is itself a form of learning that depends on synaptic plasticity. The connections within the striatum are being remodeled through practice. Dopamine plays a role here too. It's not just about reward. It's about reinforcing the correct sequence of actions, strengthening the synaptic pathways that represent the successful motor program while pruning away the ones that led to errors. This is why practice needs to be somewhat rewarding, or at least effortful and attentive, to be effective. Mindless repetition without engagement doesn't produce the same quality of learning because the dopamine-mediated reinforcement signal isn't as strong. There's a wonderful concept in skill learning research called the power law of practice. It was formally described in the 1980s by Alan Newell and Paul Rosenblum, though the phenomenon had been noticed much earlier. The idea is simple but important. When you plot improvement in a skill against the amount of practice, you get a curve that rises steeply at first and then gradually flattens out. Your biggest gains come early. The first hour of practice produces more improvement than the 100th hour. But you never truly stop improving. The curve never goes completely flat. It just gets shallower and shallower. This matches what's happening in the brain. Early learning involves large-scale reorganization, recruiting the right circuits, establishing the basic motor programs, figuring out the general strategy. That produces big, noticeable improvements. Later learning involves fine-tuning, subtle adjustments to timing, force, coordination that produce smaller, but still real gains. And the neural mechanisms shift too. Early learning is more dependent on the hippocampus and prefrontal cortex. Later learning relies more on the striatum, the cerebellum, and primary motor cortex. Ah, the cerebellum. We should talk about the cerebellum because it's one of the most underappreciated structures in the brain when it comes to learning. It sits at the back of your head, tucked underneath the cerebral cortex, and it contains more neurons than the entire rest of the brain combined. Something like 50 to 80 billion neurons packed into a structure the size of your fist. For a long time, people thought the cerebellum was just about motor coordination, keeping your movements smooth and accurate, maintaining your balance, and it certainly does that. Damage to the cerebellum makes movements jerky and imprecise, a condition called ataxia. But the cerebellum is also a learning machine. It's where you learn to calibrate your movements through practice, to fine-tune the timing and accuracy of your motor output based on error feedback. The key circuit in cerebellar learning involves climbing fibers and parallel fibers converging on cells called Purkinje cells. These enormous, elaborately branching neurons that are among the most beautiful cells in the entire body. Kajal drew them, actually, in those gorgeous illustrations we talked about earlier. The climbing fibers carry error signals. They fire when your movement doesn't match what was intended, when there's a mismatch between the predicted sensory outcome and the actual outcome. Sound familiar? It's another prediction error, just like Schultz's dopamine signal, but operating in the motor domain rather than the reward domain. When a climbing fiber fires, it triggers a form of synaptic depression, LTD, at the parallel fiber synapses on that same Purkinje cell. This weakens the connection that contributed to the erroneous movement, effectively correcting the motor program for next time. It's learning by error correction, implemented in a beautifully specific circuit. David Marr, the same David Marr who proposed the two-stage model of memory consolidation involving the hippocampus and neocortex, actually proposed a theory of cerebellar learning back in 1969, when he was barely into his 20s. And independently, a few years later, James Albus proposed a similar model. The Marr-Albus theory of cerebellar learning was one of the first computational theories of learning in a specific brain circuit, and it's held up remarkably well. The basic idea, that climbing fibers carry error signals that modify parallel fiber synapses on Purkinje cells, was confirmed experimentally by Masao Ito and others through decades of painstaking work in the 1970s, 80s, and 90s. Ito, working in Tokyo, demonstrated the LTD at parallel fiber Purkinje cell synapses and showed it depended on the conjunction of climbing fiber and parallel fiber activity, just as the theory predicted. So now you've got this picture emerging of the brain as a collection of interconnected learning systems, each specialized for different aspects of the problem. The hippocampus handles rapid, episodic, one-shot learning, the kind where you remember a specific event from a single experience. The neocortex handles slow, statistical, semantic learning, extracting general patterns and knowledge over many experiences, with sleep helping to transfer information from hippocampus to cortex. The striatum handles habit learning and action selection, chunking sequences into automatic routines, guided by dopamine reward signals. The cerebellum handles motor calibration, fine-tuning movements through error correction. The amygdala, which we haven't talked about much but which sits deep in the temporal lobe, handles emotional learning, particularly fear conditioning, stamping certain experiences with emotional significance so you remember them vividly and react to similar situations in the future. And all of these systems are running simultaneously, all the time, often learning different things from the same experience. When you burn your hand on a hot stove, your hippocampus records the episodic memory of the event, where you were, what the kitchen looked like, who was there. Your amygdala learns the fear association. Stoves are dangerous. Pain is coming. Your motor cortex and cerebellum adjust your reflexive withdrawal movement. Your prefrontal cortex updates your behavioral strategy. Check whether the burner is on before touching it. And over time, with enough experience, your neocortex extracts the general principle: hot things burn. All from one moment. Multiple learning systems, multiple types of synaptic plasticity, multiple brain regions, all working in parallel. But here's what's really fascinating. These systems don't always agree. Sometimes they actually compete with each other, and the tension between them explains some of the strangest quirks of human behavior. There's a classic demonstration of this that involves a simple card game, and it's one of the most elegant experiments in all of neuroscience. It was designed by Antoine Béchara and his colleagues in the lab of Antonio Damasio at the University of Iowa, and it was published in 1994. The setup is deceptively simple. You sit in front of 4 decks of cards. Let's call them A, B, C, and D. Each time you flip a card, you either win some money or lose some money. Your job is just to make as much money as possible. What you don't know at the start is that the decks are rigged. Decks A and B are bad decks. They give you big payouts, sometimes $100 at a time, but they also hit you with devastating losses. Over the long run, you lose money playing from those decks. Decks C and D are good decks. The payouts are smaller, maybe $50, but the losses are much smaller too, and over time, you come out ahead. Now here's what's remarkable. Most healthy people, after flipping maybe 40 or 50 cards, start gravitating toward the good decks. They develop a feeling, a gut sense, that something is off about decks A and B. And if you ask them why they're avoiding those decks, they can't really explain it. They'll say something vague like, "I don't know, those ones just feel bad." They haven't consciously figured out the math. They haven't sat there calculating expected values, but their behavior has already shifted. And here's the kicker. Damasio's team measured skin conductance responses, those tiny changes in how much your palms sweat, which are a reliable indicator of emotional arousal. And they found that before the participants consciously knew the decks were bad, their bodies were already reacting. When they reached for deck A or B, their palms would start sweating slightly, generating what Damasio called a somatic marker, a bodily signal that something risky was happening. The emotional learning system, driven largely by the amygdala and the ventromedial prefrontal cortex, had figured out the pattern before the conscious, declarative system had caught up. The body knew before the mind did. But here's where the competition between systems gets really interesting. Damasio also tested patients who had damage to the ventromedial prefrontal cortex, the region that connects emotional signals to decision-making. These patients could tell you, if you asked them, that decks A and B were the bad ones. Their declarative, hippocampal learning system had figured it out. They knew the answer. But they kept choosing the bad decks anyway. Their behavior didn't change. Without the emotional somatic marker system feeding gut feelings into their decision-making process, knowing the right answer wasn't enough to actually do the right thing. Think about what that means. You can have one learning system that has correctly identified the pattern and another learning system that's supposed to translate that knowledge into action. And if the second system is broken, the knowledge just sits there, inert. It's like having a map but no legs. This is a profound insight into how learning actually works in the real world, because it tells you that learning isn't one thing. It's not just acquiring information. It's the coordination of multiple systems, each contributing a different piece: the knowledge, the emotional weight, the habitual response, the motor execution. And when those systems are in alignment, you get smooth, adaptive behavior. But when they're in conflict, you get all sorts of strange outcomes. You get the person who knows smoking is bad, but can't stop. You get the student who understands the math, but freezes on the exam. You get the athlete who can perform perfectly in practice, but chokes in competition. These aren't failures of knowledge. They're failures of coordination between learning systems. And this brings us to something that researchers have been increasingly interested in over the last couple of decades, which is how the brain manages these competing systems, how it arbitrates between them. Because if you have a habit system that wants to do one thing and a goal-directed system that wants to do another, something has to break the tie. One of the most influential frameworks for thinking about this comes from the work of Nathaniel Daw, now at the University of Pennsylvania, along with Peter Dayan and others. In a landmark 2005 paper, Daw proposed that the brain essentially runs two parallel decision-making algorithms. One is model-free. That's your habit system, the striatal system we talked about earlier, the one that learns by trial and error, stamping in stimulus-response associations through dopamine reinforcement. It's fast, efficient, and automatic, but it's inflexible. It doesn't adapt quickly when the rules change. The other algorithm is model-based. That's your goal-directed system, relying more on the prefrontal cortex and hippocampus, building an internal model of how the world works and using that model to simulate possible outcomes before choosing an action. It's flexible and smart, but it's slow and computationally expensive. It takes effort. It takes working memory. It takes attention. And Daw's insight was that the brain doesn't just pick one system or the other. It blends them together, weighting each system's contribution based on how reliable that system's predictions have been recently. If the model-based system has been making good predictions, the brain leans more heavily on it. If the environment is stable and familiar and the habit system has been doing fine, the brain lets the habits run on autopilot and saves the prefrontal cortex for other things. It's an elegant solution, and it maps beautifully onto everyday experience. Think about driving a car. When you first learned, everything was model-based. You were consciously thinking about every action, checking mirrors deliberately, calculating when to brake. It was exhausting. But after thousands of hours of practice, most of that shifted to the habit system. You drive on autopilot, your striatum and cerebellum handling the routine, freeing up your prefrontal cortex to think about dinner or listen to a podcast. But then something unexpected happens. A deer jumps into the road, or you're driving in a foreign country where they drive on the other side, and suddenly the model-based system snaps back online. You're alert, deliberate, effortful again. The brain dynamically shifts the balance. Now, this framework also helps explain why stress and fatigue have such a powerful effect on behavior. The model-based system, the flexible, thoughtful one, is metabolically expensive. It depends heavily on prefrontal cortex function, and the prefrontal cortex is exquisitely sensitive to stress hormones, particularly cortisol. When you're stressed, sleep-deprived, or mentally exhausted, prefrontal function degrades, and the brain shifts its weighting toward the model-free habit system. This is why you reach for the cigarette when you're stressed, even though you decided to quit. It's why you eat junk food when you're tired, even though you plan to eat well. It's not weakness of character. It's a predictable consequence of how the brain arbitrates between learning systems under resource constraints. The habits were always there, running in the background, encoded in the striatum. Normally, the prefrontal cortex overrides them with better, more flexible plans. But when prefrontal resources are depleted, the habits win, and this connects beautifully to something we talked about earlier: sleep. Remember how sleep is critical for memory consolidation, for replaying hippocampal memories and transferring them to the neocortex? Well, sleep deprivation doesn't just impair memory consolidation, it also impairs prefrontal function. Matthew Walker's lab at UC Berkeley, along with many others, has shown that even one night of poor sleep significantly reduces prefrontal cortex activity, impairing decision-making, emotional regulation, and the ability to override habitual responses. So sleep isn't just important for learning in the sense of storing new information. It's important for learning in the sense of being able to use what you've learned. Without adequate sleep, the brain's arbitration between systems tilts toward rigid habits and away from flexible, goal-directed behavior. Everything connects. So let's step back for a moment and appreciate the picture we've been building. The brain doesn't learn the way a computer stores a file. It learns through this constantly shifting, dynamically balanced collaboration and competition among multiple specialized systems, each with its own type of plasticity, its own neural substrate, its own strengths and weaknesses. And the quality of that learning, how well you actually absorb and use new information, depends not just on the experience itself, but on your emotional state, your stress level, your sleep, your attention, your motivation, the neurochemical environment in your brain at the moment of encoding. All of these factors modulate synaptic plasticity in different ways, turning the dial up or down on how strongly a given experience gets written into your neural circuits. Which brings us to a question that might have been simmering in the back of your mind this whole time, one that has enormous practical implications. Can we actually improve how the brain learns? Not through drugs or surgery, but through understanding the principles well enough to work with the brain's natural systems rather than against them? And the answer, it turns out, is yes. And the science behind it is some of the most practically useful neuroscience ever produced. There's a researcher named Henry Roediger at Washington University in St. Louis who, along with his colleague Jeffrey Karpik, published a series of studies starting around 2006 that fundamentally changed how we understand the relationship between testing and learning. And what they found was so counterintuitive that it took years for the educational establishment to fully absorb it, because the conventional wisdom, for as long as anyone could remember, was that testing is a way to measure learning. You study, you absorb information, and then you take a test to find out how much stuck. The test is the thermometer, not the medicine. But Roediger and Karpik showed something different. They showed that the act of retrieving information from memory, the struggle of trying to remember something, is itself one of the most powerful learning events the brain can experience. More powerful, in many cases, than additional study time. Here's how one of their classic experiments worked. They had students read a prose passage, just a short piece of text about some scientific topic. Then they split the students into groups. One group studied the passage again and again, multiple rereadings. The other group read it once and then was asked to recall as much as they could. Just write down everything you remember, no notes, no peeking. When they tested everyone 5 minutes later, the group that had reread the material multiple times did a little better. Which makes sense, right? They'd spent more time with the material. But here's where it gets interesting. When they tested everyone again a week later, the group that had practiced retrieving, the ones who'd been tested instead of restudying, dramatically outperformed the rereading group. And we're not talking about a small difference. The retrieval practice group remembered something like 50% more material after a week. Think about what that means. The students who felt like they were learning more, the ones who kept rereading and felt the material becoming more and more familiar, actually retained less over time than the students who struggled to pull the information out of memory. The effort of retrieval, that feeling of reaching for something and not quite being able to grasp it, that uncomfortable moment of, I know I read this, but what was it exactly? That's not a sign of failure. That's the learning happening. That's the signal that's driving synaptic consolidation. And when you think about it in terms of everything we've been talking about, the molecular machinery of long-term potentiation, the protein synthesis, the CREB activation, the hippocampal replay during sleep, it starts to make beautiful mechanistic sense. When you reread something, you're giving your brain a recognition signal. The information feels familiar. Your neural circuits fire in a pattern that loosely matches the original encoding, and your brain essentially says, yeah, I've seen this before. But that recognition doesn't require the same kind of deep reactivation that retrieval does. When you try to recall something from scratch, your brain has to reconstruct the pattern. It has to trace the pathway through your hippocampal networks, reactivate the cortical representations, and essentially refire the circuit without the external cue there to guide it. And that effortful reactivation, that's what triggers the molecular cascades that strengthen the synaptic connections. It's like the difference between watching someone else lift a weight and lifting it yourself. Only one of those makes you stronger. Roediger called this the testing effect, and it's one of the most robust findings in all of cognitive psychology. It's been replicated hundreds of times across different types of material, different age groups, different educational contexts, and it dovetails beautifully with another principle that's been known since the late 19th century, but is still routinely ignored by students everywhere: the spacing effect. This one goes all the way back to Hermann Ebbinghaus, the German psychologist who, in 1885, published his groundbreaking work on memory. Ebbinghaus was a remarkable character. He essentially invented the experimental study of memory by using himself as the subject. He memorized lists of nonsense syllables, meaningless combinations like DAX and BUP and ZOL, and then tested his own retention at various intervals. Hundreds and hundreds of lists over months and months with meticulous record keeping. And one of the things he discovered was what he called the spacing effect. That information studied in spaced intervals, a little today, a little tomorrow, a little next week, was retained far better than the same total amount of study crammed into a single session. And again, this makes sense when you think about the neuroscience. Each time you revisit material after a gap, You're giving your brain another opportunity to reactivate and reconsolidate those synaptic connections. And if you combine spacing with retrieval practice, so you're not just rereading at intervals, you're actively trying to recall at intervals, you get this compounding effect that is genuinely powerful. The memory becomes more durable, more flexible, more resistant to forgetting with each cycle of effortful retrieval and reconsolidation. There's a concept called desirable difficulty that was articulated by Robert Bjork at UCLA, and it captures this beautifully. Bjork pointed out that conditions which make learning feel harder in the moment often produce better long-term retention. Spacing is harder than massing. Retrieval is harder than rereading. Interleaving different topics is harder than blocking them together. Generating an answer before being told it is harder than just reading the answer. All of these strategies feel less fluent, less smooth, less comfortable, and they all produce superior learning. The brain, it turns out, doesn't strengthen connections in response to ease. It strengthens them in response to challenge. The struggle is the signal. Now, here's where this connects back to something we talked about earlier: the dopamine system. Remember Wolfram Schultz's prediction error signal? The dopamine neurons that fire not when you get a reward, but when you get an unexpected reward, or when something surprising happens. That prediction error signal is, in a very real sense, the brain's way of flagging moments that contain new information. Moments where the world didn't match your expectations, and therefore your internal model needs updating. When you try to retrieve something and succeed after effort, that's a small positive prediction error. When you try and fail, that's a negative prediction error. But it's still an error signal, and it still drives learning, especially if you then get the correct answer. The mismatch between what you expected to remember and what you actually could remember creates exactly the kind of signal that the brain's plasticity machinery is designed to respond to. And this connects to attention too. When something is effortful, you pay more attention to it. And attention, at the neural level, involves the release of acetylcholine and norepinephrine, neuromodulators that directly enhance synaptic plasticity in the cortex and hippocampus. Michael Merzenich, the neuroscientist who did pioneering work on cortical plasticity in the 1980s and 90s, showed that cortical map reorganization, the brain physically rewiring itself, only happens when the animal is paying attention to the relevant stimulus. If the same stimulus is delivered while the animal is distracted or anesthetized, no plasticity occurs. The gates of plasticity are controlled by attention, and attention is driven by novelty, effort, and emotional relevance. So when you sit down to study something and you reread your notes passively while half watching something on your phone, you're essentially closing the gates. The information flows past your sensory cortex, gets a brief flicker of recognition, and fades. But when you close the book, put away the phone, and try to reconstruct what you just learned from memory, when you struggle with it, when you feel that uncomfortable uncertainty, you're opening every gate the brain has. Dopamine, acetylcholine, norepinephrine, all flowing, all modulating synaptic plasticity, all saying, this matters, encode this, strengthen these connections. And then, of course, you sleep. And during sleep, the hippocampus replays those effortfully encoded memories, consolidating them into cortical networks, integrating them with what you already know. The whole system works together. The encoding during waking, modulated by attention and effort and emotion, followed by the offline consolidation during sleep, followed by the retrieval practice that reactivates and further strengthens the traces. It's a cycle, and every component matters. There's a beautiful study by Jessica Payne and colleagues, published around 2012, that showed this interaction directly. They had people learn emotional and neutral information either in the morning or in the evening, then tested them 12 hours later, either after a day of being awake or after a night of sleep. The sleep group showed better retention overall, but especially for the emotional material. And when they looked at the specific sleep architecture, how much time people spent in different sleep stages, they found that the amount of REM sleep correlated with the consolidation of emotional memories, while slow-wave sleep was more associated with declarative, factual memory consolidation. Different types of learning consolidated by different phases of sleep. The brain isn't just filing things away randomly during the night. It's running different consolidation programs for different types of information. And this brings up something that I think is genuinely profound about the brain's learning systems, something that's easy to miss when you're focused on the individual mechanisms. The brain doesn't just learn facts. It learns patterns. It extracts regularities. It builds models. During sleep, the hippocampus doesn't just replay individual experiences. It replays them in compressed, sometimes rearranged sequences, and the cortex extracts the statistical structure, the commonalities, the gist. This is how you go from memorizing individual examples to understanding the underlying principle. This is how insight happens. There's a lovely experiment by Wagner and colleagues from 2004 that demonstrated this. They gave people a math task that had a hidden shortcut, a pattern in the number sequences that, if you noticed it, would let you solve the problems much faster. After initial training, some subjects slept and some stayed awake. When they came back to the task, the subjects who had slept were more than twice as likely to discover the hidden shortcut. Sleep hadn't just consolidated the individual problem-solving steps. It had allowed the brain to reorganize the information and extract the deeper structure. Insight, literally sleeping on a problem and waking up with the answer. It sounds like folk wisdom, but there's real neuroscience behind it, and it's happening in the interplay between hippocampal replay and cortical integration during sleep. So let's think about what all of this means for how we actually structure learning in the real world. Because here's the uncomfortable truth. Almost everything about how traditional education is organized runs counter to what the neuroscience tells us about how the brain actually learns best. Massed practice instead of spaced practice, passive rereading instead of active retrieval, late-night cramming that sacrifices sleep, the very process that consolidates memories in order to squeeze in more study time. Standardized schedules that ignore the fact that adolescent circadian rhythms shift later, meaning teenagers are being asked to learn complex material at 7:30 in the morning when their brains are still essentially in sleep mode. High-stakes testing that creates anxiety, which elevates cortisol, which impairs hippocampal function, and prefrontal control, which degrades exactly the kind of flexible, goal-directed learning that the tests are supposed to measure. It's almost as if the system were designed by someone who had never heard of any of this research. And to be fair, much of this research is relatively recent. The testing effect studies, the sleep consolidation work, the dopamine prediction error framework, the model-free versus model-based arbitration. These are mostly 21st century findings, or at least 21st century syntheses. The educational system was built on older intuitions, and institutions change slowly. But the gap between what we know about how the brain learns and how we actually structure learning environments is enormous, and closing that gap is one of the most important practical challenges in applied neuroscience. There are people working on this, Researchers like Pooja Agarwal, who has spent years translating retrieval practice research into classroom-ready tools. Organizations like the Learning Scientists, who distill cognitive psychology findings into practical strategies for teachers and students. And increasingly, adaptive learning technologies that try to implement spacing and retrieval algorithms, essentially software that tracks what you know and what you're starting to forget, and serves up retrieval opportunities at optimal intervals. The most well-known version of this is spaced repetition software, which goes back to Sebastian Leitner's flashcard system from the 1970s, but has been refined with increasingly sophisticated algorithms. The idea is simple. When you get a card right, the interval before you see it again gets longer. When you get it wrong, the interval shrinks. Over time, the system converges on a schedule that keeps each piece of information just barely alive in your memory, forcing retrieval at the moment of maximum desirable difficulty. And it works. The evidence for spaced repetition is overwhelming. Medical students use it to learn thousands of anatomical terms and drug interactions. Language learners use it to build vocabulary. It's one of the clearest cases where understanding the neuroscience of learning has led directly to a practical tool that genuinely helps people learn better. But it's also limited because it works best for discrete facts, things that can be put on a flashcard. The deeper kinds of learning, the pattern extraction, the model building, the kind of flexible understanding that lets you apply knowledge in novel situations, that requires something more. It requires the kind of rich, contextual, emotionally engaged encoding that we've been talking about throughout this whole journey. It requires the interplay of all those systems working together. And that brings us to something that I think is the most fascinating frontier in all of learning science right now: the question of how the brain builds and updates its internal models of the world. Not just individual memories, not just habits, not just emotional associations, but coherent, structured representations that let you simulate possibilities, make predictions, and understand how the world actually works at a deep, intuitive level. Think about what it means to truly understand something, not just recall a fact about it, but really get it. When you understand how an engine works, you're not just storing a list of parts. You're holding a mental model, a kind of internal simulation where pistons move, fuel ignites, crankshafts turn, and you can mentally trace the flow of energy from combustion to motion. You can predict what would happen if one part failed. You can reason about engines you've never seen before because your model captures the underlying structure, not just the surface details. That kind of understanding, that's what cognitive scientists sometimes call a mental model or a schema, and it's arguably the highest form of learning the brain does. And here's the thing. We're only just beginning to understand how the brain pulls this off. The neuroscience of fact memory, of habit formation, of emotional conditioning, those are relatively well mapped at this point. We've traced the circuits, identified the molecules, built computational models that match the data pretty well. But the question of how the brain constructs rich, flexible, generative models of the world, that's still wide open. And it's where some of the most exciting work in neuroscience and artificial intelligence is converging right now. Let's talk about what we do know. One of the key ideas comes from a framework that's been around in various forms for over a century, but has really gained traction in the last couple of decades. It goes by different names: predictive processing, predictive coding, the Bayesian brain hypothesis. But the core insight is beautifully simple. The brain, according to this view, is fundamentally a prediction machine. It's not sitting there passively waiting for information to arrive from your senses and then reacting to it. Instead, it's constantly generating predictions about what's going to happen next, what you're going to see, hear, feel, taste, and then comparing those predictions against what actually arrives. When the prediction matches reality, everything hums along smoothly. When there's a mismatch, when something surprises you, that prediction error signal becomes the thing the brain pays attention to, the thing it learns from. Now, if that sounds familiar, it should, because we've already encountered this idea. Remember Wolfram Schultz and his dopamine neurons? Those neurons that fired not when the monkey got the juice, but when the juice was unexpected. That's a prediction error. The whole temporal difference learning framework we talked about, that's a specific case of this broader principle. What predictive processing does is take that same logic and scale it up to the entire brain, to every level of perception and cognition. The modern version of this idea owes a lot to Carl Friston, a neuroscientist at University College London, who has been developing what he calls the free energy principle since around 2006. Friston's work is notoriously dense. Even other neuroscientists sometimes joke that understanding Friston's papers is itself a significant cognitive achievement. But the underlying intuition is actually quite elegant. The brain, Friston argues, is always trying to minimize surprise. Not surprise in the everyday sense of the word, but in the information theoretic sense. The difference between what the brain expects and what it encounters. It does this in two ways. It can update its internal model to better predict the world. That's learning. Or it can act on the world to make the world conform to its predictions. That's behavior. You feel cold, which violates your prediction that your body should be warm, so you put on a sweater. Prediction error minimized. You encounter an unfamiliar word in a sentence, which violates your prediction about what should come next, so your brain works harder to integrate it, updates its language model slightly, and next time, that word is a little less surprising. That's learning too. What makes this framework so powerful is that it offers a unified account of perception, action, and learning all in one package. And it suggests that the brain's internal models aren't just passive recordings of past experience. They're active, generative structures that are constantly running, constantly making predictions, constantly being refined. Your model of the world is alive in a way that a flashcard deck never could be. So how does the brain actually build these models? This is where things get really interesting, and where the hippocampus comes back into the story in a surprising way. We talked earlier about the hippocampus as a memory system, the place where new episodic memories are formed before being gradually consolidated into the cortex during sleep. But there's growing evidence that the hippocampus does something even more fundamental than just storing memories. It builds relational maps. This idea goes back to John O'Keefe's discovery of place cells in the early 1970s. O'Keefe, working at University College London, found that individual neurons in the hippocampus of rats would fire when the animal was in a specific location in its environment. One cell might fire when the rat was in the northwest corner of a maze, another when it was near the food dish, another when it was by the door. The hippocampus, it seemed, was constructing an internal map of space. Then, decades later, May-Britt Moser and Edvard Moser, working in Norway, discovered grid cells in the entorhinal cortex, neurons that fire in beautiful hexagonal patterns as an animal moves through space, creating a kind of coordinate system that the hippocampus can use to build its maps. O'Keefe and the Mosers shared the Nobel Prize in Physiology or Medicine in 2014 for this work, and it fundamentally changed how we think about what the hippocampus is actually for. Here's why this matters for learning. There's a growing body of evidence suggesting that the hippocampus doesn't just map physical space, it maps conceptual space too. When you learn about a new domain of knowledge, your brain may be constructing something like a cognitive map, where related concepts are nearby and distant concepts are far apart, where the relationships between ideas have a structure that can be navigated the same way you navigate a familiar neighborhood. Some researchers, like Darshan Kumaran and Eleanor McGuire at UCL, have found hippocampal activation when people are learning abstract relational structures, hierarchies, social networks, category systems, even when there's no spatial component at all. The hippocampus, it seems, is a general-purpose relational mapping engine. Space was just the first domain it evolved to handle. And this connects back to sleep in a beautiful way. Remember the hippocampal replay we talked about? Those sharp wave ripples during slow wave sleep, where the hippocampus replays compressed versions of the day's experiences back to the cortex. Well, here's what's really wild. Recent work has shown that during replay, the hippocampus doesn't just replay what actually happened. It also generates novel sequences, trajectories through the cognitive map that the animal never actually experienced. It's as if the sleeping brain is exploring possibilities, running simulations, filling in gaps in the map. Some researchers think this is how the brain extracts general principles from specific experiences by replaying and recombining episodes during sleep. The hippocampus helps the cortex build models that go beyond any single experience. This is almost certainly part of what was happening in Ulrich Wagner's insight study, the one where people who slept were more likely to discover the hidden shortcut in the number sequence task. Their sleeping brains weren't just consolidating the specific trials they'd practiced. They were recombining elements, finding patterns, building a model of the task's underlying structure. And when they woke up, that model was available to them as a sudden insight, a feeling that the answer was obvious, even though they couldn't quite explain how they knew. Think about what all of this means when you put it together. You've got a brain that learns at multiple levels simultaneously, strengthening individual synapses through long-term potentiation, building habits through the basal ganglia, tagging experiences with emotional significance through the amygdala and dopamine system, consolidating and reorganizing memories during sleep, and constructing rich, flexible, generative models of the world through hippocampal cortical interaction. And all of these systems are talking to each other, competing and cooperating, with neuromodulators like dopamine, acetylcholine, and norepinephrine, adjusting the balance based on what the situation demands. It's not one learning system. It's an orchestra of learning systems, each with its own strengths and timescales, and the magic is in how they work together. And here's something I find deeply comforting about all of this, especially as you're lying here, maybe drifting in and out. Your brain is learning right now, not just from the words you've been hearing. Though if any of these ideas stick, if some phrase or story surfaces in your memory tomorrow or next week, that's your consolidation systems doing their thing. But more broadly, your brain has been learning every moment of your life, every conversation, every stumble, every boring Tuesday afternoon, every flash of unexpected beauty. All of it has been feeding into these systems, refining your models, adjusting your predictions, shaping the vast, intricate web of connections that makes you who you are. You are, in a very real neurobiological sense, the sum of everything you've ever learned. And you're still learning, you'll be learning in your sleep tonight. The story we've traced, from Santiago Ramon y Cajal, peering through his microscope in the 1890s, sketching neurons with the care of an artist, to Donald Hebb writing his postulate in 1949, to Tim Bliss and Terje Lomo sending their electrical pulses through a rabbit's hippocampus in 1973, to Eric Kandel gently touching a sea slug's gill and watching a synapse change, to Wolfram Schultz's monkeys and their juice, to Matthew Walker's sleep-deprived students, to Robert Bjork's desirable difficulties and the testing effect and spaced repetition, all of it is really one long story. It's the story of humanity trying to understand how understanding itself works. And we're not done. We're probably not even halfway done. The questions that remain, how the brain builds its models, how consciousness relates to learning, how we can better align education with what the brain actually needs. These are some of the deepest questions we can ask. And people are working on them right now, tonight, in labs and offices and libraries around the world, driven by the same curiosity that drove Cajal to stay up late with his silver stain and his microscope. But you don't need to think about any of that right now. The beautiful thing about the brain's learning systems is that so much of the important work happens below the level of conscious effort. The synapses will do what they do. The hippocampus will replay what it needs to replay. The slow oscillations of deep sleep will carry today's experiences into the cortical networks, where they'll become part of your long-term understanding of the world. You don't have to try. You just have to let it happen. So let your thoughts soften. Let the details blur if they want to. Some of them will be there when you wake up. A name, a story, an image of a sea slug or a rat in a water maze or a monkey waiting for juice. Others will have faded, and that's fine too, because forgetting is part of learning. Remember? The system is working exactly as it should. Your brain has been doing this for your entire life, and it's very, very good at it. The world is full of things to learn, and you have a brain that was built across millions of years of evolution, refined by billions of synaptic adjustments across your own lifetime, specifically to learn them. That's a remarkable thing. And right now, the best thing you can do for all of those beautiful, intricate learning systems is exactly what you're already doing. Rest. Let the day settle. Let the sharp wave ripples do their work. Tomorrow the world will be there, full of prediction errors and desirable difficulties and things worth paying attention to. But that's tomorrow. Right now, there's just this, the quiet hum of a brain doing what it does best, turning experience into understanding, one synapse at a time, while you drift gently, easily, into sleep.